This lecture is a brief diversion where we'll discuss repetitive elements, what they are and why they occur, and why there's something that we are going to want to keep in mind as we discuss the read alignment and assembly problems. So a human genome sequence that we observe today is the end result of a pretty complex evolutionary process. And that process tends to introduce certain kinds of patterns into the genome. Patterns that make the genome sequence very different from the kind of sequence that you would get if you generated it randomly, like for example by flipping a coin over and over again, or by using a Python pseudorandom generator function to generate a random genome. Maybe the most striking example of this fact is that the human genome is extremely repetitive. It's far more repetitive than a random string would be. And so what do I mean by this? Well, some pretty interesting things happen to genomes over time. One thing that happens is that the genome gets invaded and infiltrated by little bits of DNA, sort of DNA interlopers, called transposable elements. And these are tiny little chunks of DNA that are capable of getting themselves incorporated into the genome, and then when conditions are right, they're also capable of copying and pasting themselves or cutting and pasting themselves throughout the genome. So this diagram shows one such example of how a cut and paste mechanism happens, how a transposable element can cut and paste itself throughout the genome, and it does this with the help of an important protein called a transposase. So this kind of thing has happened many, many times throughout evolutionary history to the human genome, to the point where about 45% of the human genome sequence is covered by transposable elements. 45% of all the bases in the human genome came from a transposable element. And there are many different kinds of transposable elements. As you can see from this pie chart here, they come in various abundances. And then there's one particularly renowned example of a transposable element, which is called alu, which is highlighted right here in this red bit of the pie chart. So alus actually occur many times in the genome, more than 1 million times, and about 11% or so of the human genome sequence is covered by these things called alus. So why should we care about these repetitive portions of the genome? Do they really affect whether we can design good algorithms for solving the read alignment problem and the assembly problem? Well, actually they do. So they do because they create ambiguity. And if there are, for example, 1 million copies of the alu element and one of our sequencing reads comes from inside one of those copies of the alu element, and then we want to solve our read alignment problem, you can imagine that's difficult. There's ambiguity. It's going to be very difficult to figure out exactly which copy of the alu repeat our sequencing read came from, or maybe even impossible. So in our so here's a illustration of this. So here is actually a picture of all the chromosomes of the human genome, and everywhere where you see glowing green on this picture, that's a place where there are alus in the genome. So they're spread all over. And if we're in a situation, let's say now this is a schematic version of our genome that has some unique sequences, some non-repetitive sequences, which are shown in blue, and it also has many copies of the same repeat. Maybe it's the alu repeat. But those copies are shown in red, and let's say we get some sequencing reads from the blue portions of the genome, and we try to solve the read alignment problem. Well, that's good, because we'll be able to resolve where they come from, because they come from non-repetitive portions of the genome. Now let's say we have a read that comes from inside one of the copies of this repetitive element in the genome, and this occurs in several copies, including the 3 that you see here. Well, we're not really going to have any idea whether the read came from the copy that it truly came from or whether it came from one of the other copies. There's just going to be inherent ambiguity in the read alignment problem in that sense. So in our puzzle analogy, you could say this is a little bit like a puzzle where half the pieces are just featureless blue sky or something like that. So that if you're trying to figure out where one of those pieces comes from, it's going to be very difficult to distinguish which part of the sky it actually comes from. And then later when we talk about assembly, we'll see some other important ways in which repetitive sequences in the genome are going to create problems for our algorithms.